Welcome to Derms on Drugs, a video podcast brought to you by scholars in medicine. Derms on Drugs is where cutting-edge derm meets so-so comedy. I'm Matt Zires, and each week I'm joined by my residency buddies Laura Ferris and Tim Patton. And we use our 60 years of combined derm experience to discuss, debate, and dissect the hottest topics in dermatology. It's everything you need to know to be on the cutting edge of derm, and it'll be the most fun you've ever had while actually learning something useful. New episodes drop every Friday, so make sure you set your calendar. And boy, do we have some cool stuff this week. So over the last, for those of you who are not like itch, live and die, love the itch research world, we've had some of the most fundamental questions in the history of dermatology answered in the last couple of months here. And I'm so excited to have Dan Kaplan from the University of Pittsburgh on to talk about his article on scratching and itching, which was published in Science. Science, for God's sakes. That's like as real as a journal gets. I personally am convinced that it is the first time that an article about scratching has ever been in Science, but I didn't look that up to confirm it. But so let's just go ahead and jump right into it. And I'm going to start just with an article that I think is fun and has been around for a long time. So this was a study out of roughly 10 years ago done by Gil Josipovich, the godfather of itch. So it's brain's reward circuits mediate itch relief, a functional MRI study of active scratching. And basically, here's what Gil did. He put people in an MRI machine and then induced, so first just scratched like an area on their forearm, and then induced itching using cowhage, which is itch powder. And then either an investigator scratched it or the person scratched it. And so the investigator scratching it is passive scratching, the person scratching it is active scratching. And he looked at what parts of your brain lit up. And the main takeaway from this was that itch relief, active scratching and itch, activates the same parts of your brain as having sex, gambling, using drugs, all these addictive behaviors. And so there is a strong, what he proved was that there's a strong pleasurability aspect to itching. And that has always fascinated me because of the idea that, like, why was itch so valuable that evolution put it on the same level as getting sugary foods, fatty foods, whatever. And we really have never had a good answer to that. And I think the papers we're going to talk about today start to give us some of that answer.
Matt, it was only scratching when it was only if you did the scratching and you were actually itchy. So it's not the scratching, it's the quelling the itch that is what creates that pleasure circuit. Is that right?
Yes.
Okay.
So that's exactly right. And interestingly, because it is an interesting question of what feels better, you scratching an itch, your own itch, or somebody scratching it for you. And this really looked like it was somebody scratching it for you. Because that's an interesting question. If you think about like an itchy spot on your back, like how good it feels whenever somebody hits the right spot.
But they never do.
Right? That's exactly right. They never do.
They never hit that exact spot.
And that was actually part of this article, because part of me wondered, did other people scratching it feel better just because they got it, just because not feel as good because they just didn't quite get it right, they weren't in the right spot, the right strength. And it turned out that whenever you scratch your own itch, it activates different parts of your brain than if somebody else scratches your itch. So there is a fundamental difference. It's fascinating. I never knew what to make of it, but it was just fascinating. Patton, you got anything you want to say about this one?
No, I think we talked about this. I think that, we scratched to get shit off our skin that was going to be bad for us. And I know you talk about mosquito bites and that doesn't happen until later and how ticks don't itch. But if you think about a flea infestation or louse or mites or lice, you got to get that shit off you or it's going to probably give you some bad disease. So I was happy with that explanation. I'm not saying that these other explanations are wrong. I was good with that one.
Okay. I still say, man, that by the time it's itchy, the bug is gone from the spot that is
But
itchy.
if you think about fleas in general,
Fleas.
though, right? We're prehistoric little mammals. We're covered in fleas, and like they start biting you, and you start to get itchy, but they're still on you, and so you just gotta scratch them off.
But they're not in the spot that's
No,
itching. If like
but
right here is
What's
itching,
that?
it does
then the
matter.
flea was there yesterday.
Right, because it takes a month to get the adaptive response, right, to fleas, and like scabies takes about a month, right, before you
Yeah.
start to get itchy. And if you think about people who can't scratch, like in a nursing home or something, they get Norwegian
Right.
scabies, right? So the scratching really does have a benefit to removing mites, scabies, but it's probably the same for body
Yeah.
lice.
Damn it,
Yeah.
Dan! It's hard for me.
Okay, we're gonna, yeah, no more
Norwegian
smart people.
scabies.
No more smart people on our,
Ah,
we don't
I
sound
don't have an answer to
Thank you,
that.
Dan.
Yeah.
Damn it, Pat's been trying to convince me for a week, and I'm like, nope, nope, nope. You got me with right there. Norwegian scabies in nursing home patients.
All right.
Right, but in the case of like ticks or mosquitoes, you'd argue that, well, they've evolved to actually avoid the immediate
I
itch
was gonna say
response,
the same
right?
thing. Yeah.
Otherwise,
Like,
they get otherwise they'll be taken out.
and I
right.
think maybe that's why ticks evolved to like there were probably ticks that did induce that itch right away, but you start scratching them off, and maybe it got rid of it.
All right, let's move on to our next article here. So, Dr. Patton, what do you got?
Well, I did this one from January 2025 JID entitled Epidermal Mechanical Scratching Induced ROS, so reactive oxygen species, exacerbates the itch scratch cycle via TRPA1 activation on mast cells in atopic dermatitis by Hu et al. A long title, but that's really the takeaway. You scratch, keratinocytes make reactive oxygen species, ROS activate mast cells via TRPA1. Mast cells release all their things, and you get more itchy. And so they did it with a bunch of experiments. I don't, the first experiment they stretched these keratinocyte cell lines, and that made keratinocytes release reactive oxygen species that was suppressed by N-acetylcysteine. Second experiment, they irritated mouse skin with a chemical and tape stripping, and they showed that you have increased mast cells and tryptase in the dermis. Tape stripping, which did higher levels of both. They took this mast cell line. They showed if you stretch the keratinocytes, you take the supernatant, you add it to a matrix thingy that they made with the LAD2 cells, which are this mast cell line. It makes these mast cells release tryptase similar to if you just exposed LAD2 cells to H2O2. They did gene regulations, 945 genes upregulated, 654 genes downregulated. I think we should go through each one of these for completeness sake. No, they did cluster analysis on those genes. And the summary from the gene studies was upregulation seen in genes involved in mast cell activation, type 2 inflammation, redox processes, ion channels. I don't, there were 4 other experiments. I don't really feel like going all through the whole things. This is all Cyrus's fault. He was like, hey, you should do this paper. And I was like, yeah. And then I went to print it out, and it was like 47 pages of stuff. It was really hard. You needed a PhD to fully understand everything. But the
So
takeaway,
what's the
the
yeah?
takeaway
What's the
was the
takeaway?
title,
That's
right?
the key thing.
Stretched Hackcat supernatant appeared TRPA1 expression on mast cells. That led to the release of tryptase. So that's why we itched. And it also had these other pro-inflammatory cytokines. I'm guessing, Dan, you understood this paper way better than I did.
Yeah,
What were your
I thought
takeaways?
I thought it was a really interesting paper. I guess it's not out yet, so I'm glad you sent it to me. I like reading
Yeah,
papers in the
we're on the
JID.
cutting edge
So
on
it
this
like
show.
Cutting
always,
edge.
always. And so what it does is it adds to the list of things that keratinocytes release, things that keratinocytes can release that are itchy, right? So when you scratch the skin, you release one cytokine called TSLP. It's very well characterized, and it's very, it can activate mast cells and other cells as well, right? And can be important in triggering itch. And IL-33 is another example of things that can be released. So it's a really nice paper to add to the list of the complex stuff, I'll use the word stuff, that is released by keratinocytes that can have biological effects on mast cells and on many other cells in the skin as well. So I thought it was really. Really, really interesting for that reason.
What, my takeaway from it was that they made this gel and put keratinocytes in it, and then they went basically and showed that moving the keratinocytes, like deforming them just a little bit, generated these reactive oxygen species that then, activated mast cells and the whole thing. Because I've always had a big problem with the itch scratch cycle. Like, people like to talk about the itch scratch cycle, and this finally made me buy into it. And the reason I never really bought into it was that if it was just that the scratching is mechanically damaging the skin surface, then in all of our atopic patients, it ought to be that we can see manifestations of scratching everywhere that they've got a rash. And you can't. The majority of places where they've got a rash, there's no evidence of scratching. So I've always been like, that's baloney. But this is some of the first stuff in Dan's paper as well that really says to me scratching itself might have a way that it's generating inflammation.
Yeah, a good
And
example.
the
Good
other thing
example
that made
of
me
that
think about is,
is
go
is
ahead, Dan.
is people who are dermatographic, right? That's a perfect example of it, where now you've got mast cells that are not fully activated, but they sort of have this heightened reactivity. And
Yeah,
now, if you scratch the skin, the stimuli from scratching is sufficient to push those mast cells over the edge, degranulate, and you get urticaria wherever the scratching has occurred.
That's interesting. So yeah, so it's really, and it's not like you're scratching and excoriating. It's really just the pressure of the scratching, and it's
Yeah.
the yeah. So I like the idea from this paper that it's like the mechanical stretching of the keratinocyte. That's the signal. The other thing I thought was interesting is that N-acetylcysteine inhibited this, and we know that there's some data that N-acetylcysteine can also be helpful in like skin picking disorders, right? So maybe it sort of squelches the reactive oxygen species and the downstream effects. I don't know. I mean, I always kind of wondered, why does N-acetylcysteine work? Do you think it fits into this mechanism, or do you think it not really?
That was the last experiments they did were, yeah, let's squelch or whatever, gather up these reactive oxygen species with N-acetylcysteine. They also had a medication that blocked that TRPA1. I think it's like a channel on the mast cells. And so were both of those, and doing them combined was better than each one on its own. And so, potential therapeutic for itchy stuff is the TRPA1 blocker along with something that sort of quenches the reactive oxygen species.
I'm going to put in a quick plug for Vitama here, because Vitama tapinarof actually has data that it is a direct the molecule itself is a strong antioxidant. In addition to upregulating endogenous antioxidant pathways, and every time they've brought that up, I've been like, "You guys are out of your minds. ROSs have nothing to do with atopic dermatitis. This is baloney. Don't even talk about it. Blah blah blah." And son of a bitch, it turns out I was
And
wrong, which is
they
very
pay you for this
And yeah,
advice.
yeah, we don't have disclosures on drugs on dermatology, but yes, derms on drugs. I mean, so yeah,
Worth every
if we
penny.
well.
did exactly.
Wait, I got one other thing I got to ask what you guys think about this. So the other thing that the stretching made me think about this, and Ferris, I don't know if you remember, this was a discussion we had in residency. It's always fascinated me that psoriasis and atopic dermatitis happen in areas that have the most skin stretching, right? The places where your skin is most actively deformed, right? Crooks of your elbows, backs of your knees, extensors of those areas. And we had a conversation where I was like, do you think it could be like the mechanical stretching of your skin that is doing this? And your answer was basically, oh, who the hell knows? Like, I don't know how, but
That was my
maybe
answer
this
to
is
most
part of,
things in residency, but yeah. I mean, yeah,
but
I
maybe
also
this is,
think that there's probably like, why is one extensor surfaces? I think that there's probably something to like trauma, right? To elbows and knees that's different than maybe
yes,
the stretching. But yeah, all right.
but
Well, maybe we'll figure
for
this all out
flexors,
today.
I always thought that it was the, what the hell do you call it? The sweating in your flexures, but maybe it's both the sweating and this.
Right.
But let's go ahead and move on here. Dan, why don't you? So I'm a basic science idiot. I have no idea what happened. Patton's less of an idiot about basic science than I am. Ferris is actually smart. So can you explain your article in a way that I would understand?
I think you undersell yourself, but I will try. So the lab for a long time has been interested in how neurons in the skin are able to control or affect inflammation in the skin. And so we've been focusing on these sensory neurons in the skin because, of course, there's all different types of neurons, right? And so some of the neurons can sense like painful stimuli. This would be like heat, and they express a receptor that it's called TRPV1, TRPV1. And that's actually the receptor for capsaicin in chili peppers, right? And so these are neurons that are activated by, by trauma, heat, things like that. And we and others have found that these neurons are really required to trigger all kinds of inflammatory pathways in the skin, whether it's like a psoriasis type or a dermatitis type, or just about anything in the skin requires those neurons.
And Dan, the latest stuff has been that immune cells kind of cluster at the tips of the sensory neurons. Am I? I
Yeah.
think I saw
So
that somewhere.
yeah, so the neurons and the sensory neurons in the skin run along the neurovascular bundles, and you can see that lots of cells are hanging out right next to them. Dendritic cells are there, and of particular interest is the mast cell, right? And they're just hanging out right along those neurons. Obviously, they're talking to one another.
Yep. Now, Dan, I'm going to say right now, when I think mast cell, I think histamine, and that's it. Right?
Yep.
That mast cell releases histamine. That's it. I know obviously at this point I've learned better that I'm wrong about that, but I think a lot of people still you think mast cell, and you think histamine. That's
Well,
it.
yeah, well, mast cells release a lot of histamine, but if you look what's inside the granules, there's like a witch's brew of different proteases and inflammatory amines, and they also make cytokines and chemokines and all kinds of things. It's this incredibly complex mixture. Histamine is part of it, and if you also think this is apropos of what we're going to talk about, if you also think about how mast cells are activated, right? Everyone is going to think about IgE, that's made by a B cell, and it's pre-bound to the antibody, the IgE receptor on the surface of mast cells, and it just hangs out in that state until an allergen comes, cross-links the IgE, and then you get mast cells degranulated, right? That's the classic way of activating a mast cell. It turns out there are many, many, many ways to activate a mast cell. And I think one of the most exciting ones is through this new type of receptor. It's called MRGPRX2. And mice have one called MRGPRB2. And so this is a receptor for many, many types of ligands. Like, for instance, some products of the microbiome on the skin can activate it. The cathelicidin, which is made in rosacea by keratinocytes, can activate it. But most importantly for this discussion is that there's this neuropeptide released by the neurons that sense pain called substance P. And substance P is a ligand for this receptor on mast cells and causes the mast cells to be activated. So the neurons, like these pain sensing neurons, when they get activated, they release substance P. And one of the things they do is they cause mast cells to get activated.
Dan, is MGPRX2, or I don't even know if I got that right, is it specific to mast cells? Because like
In
there's
mice, it is specific to mast cells. In human, it seems like it's specific to mast cells, but I will defer. There are a lot of people working on MRGPRX2 as a therapy for both urticaria and atopic dermatitis, and it's unclear if it's maybe also expressed on some types of neurons. But certainly mast cells, and it seems within the immune system and within the skin, it's pretty much on mast cells and maybe neurons in certain contexts.
So that'll be interesting because I'm doing a clinical trial right now with a drug that targets c-Kit, and that is not specific for mast cells. It also is on melanocytes, and
Right.
so you get hair whitening whenever you attack c-Kit,
So it's
and
working.
so the MGPRX2 is going to be an interesting other target.
Yeah. So I'm, that is under development. Anyhow, so that's a little bit of background. So we had been working on these pain sensing neurons, and we started to think like, well, what about itch? There are other neurons that are responsible for itch. And so we got another mouse strain from, Zhang Dong, who's at Hopkins, who's really one of the leaders in this field. And that allowed us to delete one subset of neurons that was responsible for itch. And I had this idea that, since the pain sensing neurons were required for a type 17 response, that I thought maybe the itch neurons, they would be required for a type 2 response. So we played around with that, and that, unfortunately, like so many of my great ideas, turned out not to be true. But what we did find is that if we were doing models of allergic contact dermatitis, where you put small molecules, you make the mice allergic to the molecule, and you challenge them a little bit later, that the mice that didn't have this itch sensing neuron, they wouldn't get inflammation, right, in this allergic contact dermatitis model. And what was interesting, and this was the weird part, this is what got us started, is if you did haptens that we knew required IgE and mast cells, you needed the itch neurons. But if you did a hapten that didn't seem to require IgE, it was fine. The mice were fine. And so we scratched our heads for a little while thinking like, well, what are the neurons releasing? And then we just did this super simple experiment where we did the dermatitis model, but we put the mice in Elizabethan collars. Imagine a very small cone of shame, that you have for your dog or cat. They make them from mice, and you just put the mice in this cone of shame so they can eat the drink, they're fine, they just can't scratch their ears. And it turns out that you didn't need the neurons per se, you needed the scratching. So you needed the neurons to trigger the itch sensation resulting in the scratch, and that's what you needed for the inflammation.
Dan, that's one of our sayings here on Derms on Drugs, is that one of the most important phrases in all of science is "that's weird."
Yeah.
And when you got away from the itch, you got rid of the itch neurons and the inflammation didn't happen, and you said that's weird. Then you did the new experiment. Oh my God, it's not the itch neuron, it's the scratching, but it's the tracking down the weird stuff that you don't expect that leads to big breakthroughs.
So the hard part was to figure out why is scratching required for inflammation? So we talked earlier about all the things that scratching can release in the skin, right? I mentioned TSLP, we talked about IL-33. And so we went down this list. And it's like, yeah, scratching does trigger them. When you do the dermatitis, it does trigger them. But plus or minus the Elizabethan collars, there was no difference. So that didn't seem to be, at least in this model, triggered by scratching. So it turns out that what's happening is you need the scratching to activate the pain sensing neurons. These pain sensing neurons release the substance P, remember, and that binds to the MRGPRX2 on the mast cells, and that activates them. But that's not enough, right? What really needs to happen is if you activate a mast cell using this classic IgE pathway like a regular allergen will do, you get like level 1 of mast cell activation. If you activate just with the MRGPRX2, you also get level 1 of activation. But if you activate both pathways at the same time, you get this big synergistic response. And that's what's required for the inflammation.
Wow.
And so that's why scratching was important. And so the idea then is that, so scratching is actually making the mast cells more likely to degranulate. And a way to think of this is that if you have an allergen that's maybe sort of subclinical, like it's not quite enough to trigger the mast cells, if you now start scratching the area, now it'll degranulate and you can get the inflammation going, or the protection against the helminth, or the protection against whatever it is, right? So it's like it's lowering the threshold or increasing the efficiency of mast cell activation.
So is mast cell activation through IgE versus through MGRX, whatever? Is it the same kind of activation? It doesn't does it cause different substances to be released if you trigger through one or the other?
Super cool question. So we've known about this for a while. In fact, Brian Kim has a very nice paper on this a number of years ago, and others as well before him have found that if you activate a mast cell through IgE or through this MRGPRX2, different sized granules are released, right? So that was known. And it was known that like the cytokine TNF, which is very important for inflammation, if you activate through the Fc epsilon through IgE, you'll get TNF, but MRGPRB2, you don't get a lot of TNF. Right. And so, we did this and we found that TNF was one of the main things that was different, but we have not looked properly. I'm sure if anyone like systematically really looks at it in detail, there'll be a lot of changes. And up until now, it was kind of unclear why mast cells should be releasing different stuff and what it all means.
So, Dan, Tim and I have been, or Dr. Patton and I, excuse me, even though, by the way, if anybody is watching and thinking, why does Patton just not look as good this, his computer, in addition to using an Android phone, which is just unbelievable, his computer is apparently from 1987, and it crashed
I
on
have
him.
an Android sponsorship, so I don't like to say it.
So we have been talking about if mast cells are actually involved in atopic dermatitis, because and as I've thought, and my answer and his answer up till now has been no. Like, man, because you can
Why,
put
why,
all the
why do you say
Because
that?
mast
You
cell
say that because you
because
therapies,
go there.
I always thought it because mast cell therapies didn't work, right? That's what we're talking.
Right. What mast cell therapies?
Antihistamines.
Yeah, well, antihistamines is blocking like
I know.
one of like 500
I
things
know.
to
We're
release.
dumb. We're dumb. And then I
Yeah.
said
And but
omalizumab doesn't work either, but omalizumab is IgE specific. Basically, so is your argument that there are mast cell pathways that we will discover that are going to completely change our treatment of atopic derm?
I'm saying I don't know, but I do know that there's quite a bit of pharma interest in blocking the MRGPRX2 pathway. A number of companies, some of which have been acquired by bigger companies, and there are a number of clinical trials going on. And I'm not privy to them, so I don't know if they're working. I would think it'd probably be very effective in urticaria, right, and urticarial-related disorders. And whether it also works for atopic dermatitis, I think it probably should. But I don't know the answer to that, but I bet we're going to find out pretty soon.
so Dan, if I can let me see if I can summarize how I think about this now and tell me how wrong I'm sure I'm wrong, but the question is how wrong. So because I'm always thinking from an evolutionary perspective, why would it have gone this way? And so I now think of it as being the scratching activates an immune response that is not just removing stuff from your skin, but is also, let's say that the bug that bit you put a little bacteria in there too, and you start scratching it before it turns into an infection, you're like priming your anti-infection response.
Yeah,
Is that
I think that's 100% right. So we focused up till now talking about how scratching makes more inflammation, which, I think the upshot from this first part I've talked about is that scratching makes your rash worse, makes the inflammation worse. And I think this tracks very well with everyone's experience, right? If you have eczema, you scratch it, it feels awesome, but you're going to pay for it, your rash is worse. If you have a mosquito bite and you ignore it, it usually goes away pretty quickly. But if you scratch it, you'll get, a persistent urticarial papule that can last for a week or so. So, the question we asked at this is the second half of the paper is, you get more inflammation from scratching. Is there a benefit from it? And what we found is that if you look at the microbiome of the skin of these mice, when the mice scratch, their microbiome is less diverse. Some of the bacteria are no longer present. And if you do a model, a Staph aureus model, this is just an epidermal staph, or like an impetigo model, basically, that the scratching reduces the amount of staph on the skin by a fair bit. And if you either have mice where they don't sense the itch, where you get rid of the itch sensing neuron, or if put those cones of shame on them so they itch but they can't scratch, now they have a normal level, they have a high level of staph. So the idea then is that scratching, while being detrimental if you have an allergic situation like eczema, is actually beneficial, at least in the short term, because it helps reduce the amount of Staph aureus on our skin. And I think that's one of the reasons why scratching may have evolved, may have persisted. And I think it's also one of the reasons why it feels so good to scratch and itch, because it's actually a reinforced behavior, right? It's a good thing.
Fascinating. So do you think this
That's
has a
good.
role in like bullous pemphigoid, like thinking about things that really itch? And like, do you think that the itchiness of bullous pemphigoid causes sort of out of control inflammation in older people, and that's why we get recruitment of eosinophils and blistering? Or I don't know, maybe it
That's
doesn't.
a super interesting question. I don't know.
Do you?
Yeah.
I don't know. Yeah.
So but now we think of these medications that let's get rid of itching. It's like what gets rid of itching the quickest. Um, is itching purely evolutionary? Do you think we still derive a benefit, or do you think there's any sort of potential harm of, if you could completely knock out itching, do you think that would hurt us?
Only if you had scabies.
Okay.
Right? No, itching. Okay, itching is like a useful behavior, right? No, useful sensation leading to a often useful behavior, right? So what's the point of pain, right? Pain is to let you know that you've got some trauma or something going on. You should attend to this problem, fix it, but also you should remember. It lays down memories that whatever you just did that caused that pain, don't be an idiot and don't do it again, right? But scratching is very different. It's reinforced, right? So you want to do it more and more and more. So I think they're fundamentally different.
Interesting.
It's the carrot and
All
the
right, well,
stick. Itching is
Dan,
the
this has
carrot.
been
Yeah.
a fascinating discussion that has been a ton of fun, and we're now going to move on to what it turns out is everyone's favorite portion of the show, where Dr. Patton is going to pull out his trivia machine,
Ooh,
which is really just himself.
it's not on his laptop, is it? That would be trouble.
It's not a what?
It's not on your laptop, is
No.
it?
I printed it out. I have a backup plan.
Wow, you use paper. Impressive.
Paper and printer.
All right. So, Dan, we reviewed the rules before we started. Dr. Patton, let's go.
Yeah. By the way, as I was reading your paper, I kept thinking you really need to make like if you give a mouse a cookie, but with your research. Like if you give a mouse diphtheria toxin, you'll knock out MRGPRA2 neurons. If you knock MRGPRT with like little cartoon drawings and like, I would have understood this much, much better.
Oh, there is. Did you get to supplemental figure 48? Because there is a model there.
No, like that's the thing that kills me. You had, however many experiments, maybe 6 separate experiments, and then you're reading along and it's like, oh, check supplemental. And I'm like, no, I don't understand these 6 experiments.
Well, yeah, it's just a necessary evil.
No,
the process.
It's smarter way smarter than I am. So it frustrates me. All right.
And it's just differently smart than you. Differently smart.
Thank you. My therapist, Matt. All right. So these are questions about trivia about pruritus, which that's how I say it.
Pruritus.
Don't stop me. All right.
Pruritus.
Here we go. What cartoon duo, known for their over-the-top violent antics, often entertains Bart and Lisa on The Simpsons?
Itchy and Scratchy.
That's Kaplan. By a hair.
Oh.
What 1955
Isn't
film?
that ironic that Kaplan beat me by a hair? That's all I'm going to say. Go ahead.
Oh, ouch. That's not nice. What 1955 film features Marilyn Monroe in her famous white dress scene, standing over a subway grate with her skirt billowing upward?
7-Year
The
Itch.
7-Year Itch.
Kaplan again. He's just...
What was it?
It's 7-Year Itch, but Kaplan's in earlier from where I am.
Because you're both
he's
sitting
quicker.
in Pittsburgh,
He's quicker on
I'm
the
in
buzzer.
North Carolina. It's like why they put the traders in New York. It's
you could be in Pittsburgh.
Electronic delay. All right.
All right. Third and final question: What bacterial disease caused by Bartonella henselae shares its name with a Ted Nugent song?
Cat scratch fever.
Cat fever.
Yep. Once Xyrus 043. Man.
Did Ferris beat me to that?
Yeah.
I did.
You may be on a delay. You're a little choppy today.
Okay. That's
but it's not your video.
All right. That's it.
Dan, so again, I remain on the losing streak for the entire season. So we'll stick with that.
Can I ask a question? Matt, since I had 2 out of 3, can I get your voice on my voicemail as like a prize?
Like,
Or do I get to choose any of the panelists?
wait, don't tell me type thing. Yeah.
Yeah.
Do you know what my voicemail actually says? It says, don't leave me a message. That's it. It's the whole voicemail. All right. Well, Dan, thank you for joining us. This was such a fun discussion, and I want to thank all of our listeners for joining us as well. If you got questions, comments, ideas for topics we should cover on the show, shoot us an email at questions@dermsondrugs.com. Again, that's questions@dermsondrugs.com. And I hope you learned a few things. I hope you laughed once or twice. Mostly, I hope you're planning to join us next week. So until then, I'm Matt Zyrus.
I'm Tim Patton.
And I'm Laura Ferris, and we are Derms on Drugs.